Assalamualaikum and good morning to all panels and members of the floor. I am Raihannah Syuhada and along with me today is Nurul Husna. The title for our presentation this morning will be Formulation and Evaluation of Mer Toothpaste with Associate Professor Dr. Luay Thanoon Younis as our supervisor.
The problem statement of the research is the decay of teeth and gingival problems are commonly occurring and negatively affecting the quality of life of population. Despite of having improvement in oral health measures and usage of fluoridated toothpaste, oral health problems incidence does not stop yet. Hence, there is a high demand for innovative efforts to develop a new product which may help in reducing the intensity of oral problems. According to Kareva et al. 2020, various oral healthcare products containing herbal ingredients are available in the market. Randomized control trials have shown that herbal oral care products are relatively superior to non-herbal products. And to formulate a herbal oral care product, the active ingredient must be safe, non-toxic, effective, and well-known to the public. Next, historically, myrrh was used in the Roman Empire to treat the wounded soldiers. It is also used in Chinese medicine and aromatherapy. It is a resinous exudate and is obtained from the stem of different species of Commiphora trees. Myrrh can also come in many forms such as oil, resin, and gums. The name myrrh comes from the ancient Arabic and Hebrew word mur, myrrh, which means bitterness. Myrrh helps in reducing dental plaque and gingival inflammation. It also has multi-actions of antibacterial, antifungal, anti-inflammatory, antiseptic, and astringent effects. Myrrh is reported to have medicinal properties and have been used in traditional medicines against a variety of diseases. The objective of this research is to formulate and measure the cytotoxicity level of organic myrrh toothpaste. The first methodology is the extraction of myrrh. At first, myrrh resin was crushed into powder with pestle and mortar. And then, myrrh powder was then soaked in the ionized distilled water at a ratio of 100 gram myrrh with 500 ml of water at room temperature for 48 hours. After that, the watery myrrh was filtered to obtain the purified myrrh solution. In the following, the filtration process, myrrh solution was placed in centrifuge tubes and underwent a freeze drying process for 72 hours. The freeze-dried myrrh was kept in the fridge to be used in the preparation of Mer toothpaste. The second methodology is preparation of Mer toothpaste. These are all the ingredients that were used to prepare the Mer toothpaste. First one is myrrh extract, polymer, surfactant, sodium bicarbonate, and natural sweetener. After we have done several pilot trials in measuring the ingredients quantities and mixing procedure, the following procedure was done to prepare Mer toothpaste. Firstly, 65 grams of baking soda was poured into a bowl, and 5 tablespoons of xylitol or natural sweetener were added into the bowl as well. After that, 32 grams of HPMC polymer were gradually added into the mixture. And then, 2 teaspoons of myrrh extract and a few drops of sodium lauryl sulfate were poured into the mixture. All the ingredients were mixed well until the desired texture was achieved. Besides, we also undergone cytotoxicity tests for this research. Firstly, the cells were cultured in the 96 well microplate, and then 50 microliter of complete FM and cells were added to each 96 well microplate. The cells were then incubated at 37 degrees Celsius, 5% carbon dioxide, and 95% of oxygen for 24 hours. The confluent cells were then taken from the incubator and viewed under an inverted microscope to check the percentage of the confluent cells. The medium in the 96 well microplate containing confluent cells was removed by a micropipette. After several pilot trials of cell viability tests, it is decided to use the following concentrations of 100 to 400 mg per milliliter to assess the effect of the paste on the viability of gingival fibroblasts in the test sample. While the placebo samples contain polymer only were used for treating the control samples. After that, 100 microliter of each Mer Toothpaste concentration were added into the divided wells. The cells were then incubated with Myrrh for another 24 hours. At the end of the incubation period, the entire medium was removed from the wells using a micropipette, followed by the wells washing with 30 microliter of PBS. And then 100 microliter fibroblast medium was then added for each well. MTT cell proliferation assay was implemented according to the method described by Yazawa et al. 2005. 15 microliter of dye solution was added into each well, followed by incubation for 4 hours. During the 4-hour incubation, living cells convert the tetrazolium component of the dye solution into a formazine product. After the incubation, 100 microliter of stop solution was added to each well. The experiments were performed in triplicate. The optical cell density was checked using a microplate reader at a wavelength of 570 nanometer. The cell cultures were assessed for their optical density at intervals of 24 and 48 hours treatment. As you can see in the first photo, that is the cells culture before treatment with Myrrh paste. And the next one is the cells culture after treatment with Myrrh paste. As you can see, there are more gingival fibroblast growth in the second photo.
This is a bar chart of the result after 24 hours of treatment. The obtained data were analysed by SPSS version 27. Kruskal-Wallis statistical test was used to compare the effect of Mer toothpaste concentration on fibroblast growth. Pairwise comparisons was used to compare difference in the cell growth of samples treated with Mer paste. Meanwhile, man-Whitney test was also executed to evaluate the differences between Myrrh group and placebo group after 24 and 48 hours. As we see here in this chart, Myrrh paste with concentration of 300 mg per ml is inducing the highest fibroblast viability compared to the other concentrations used. This shows the graph of the result after 48 hours. And after 48 hours, 300 mg per ml concentration was still maintaining and enhancing the viability of fibroblasts. But above that, as shown in the chart, with 400 mg per ml concentration, the cell growth has declined as compared to the other test groups at both 24 and 48 hours. Based on the chart, the results show that Myrrh paste at concentration of 100, 200 and 300 mg per ml concentrations are enhancing the growth of gingival fibroblast growth after treatment for 24 and 48 hours. However, by increasing the concentration above 300 mg per ml, the cells were apparently exhausted and showed less growth ability. Results also showed that the highest level of cell growth at 24 hours with cell viability of 938% and 48 hours with cell viability of 772% were obtained in samples treated with 300 mg per ml concentration of Myrrh paste. In this study, the results indicate that Myrrh paste concentration at 300 mg per ml is the optimal concentration, as it induced the highest cell viability as compared to other concentrations used in the trials after 24 and 48 hours. Samples treated with 400 mg per ml concentration were having less cell viability than 100, 200 and 300 mg per ml. This may imply that 400 mg per ml treated samples were receiving intolerable dose of Myrrh paste concentration that significantly suppressed the growth rate of these samples as compared to the other test samples. In the placebo samples, the cell viability remains almost constant in all concentration groups and noticeably less than that in the test samples after 24 and 48 hours. During the formulation process, it is advisable to implement a continuous stirring and use of nanoparticles Myrrh powder to ensure the homogeneous consistency of Myrrh toothpaste. As for conclusion, this research has shown that Myrrh toothpaste is safe to be used at a certain adjustable concentration. Future clinical studies are recommended to evaluate the safety and effectiveness of Myrrh toothpaste.
These are the references that we used to complete this research.
And with that, thank you.